Oh, wait. I think I shared my entire desktop. So. Is it okay on Zoom and then also in the room? Yes.
Yes.
Okay, awesome. So, hello, everybody. I met a few of you just a couple minutes ago, but I'm Hope. I'm a grad student in Manu's lab, and then I work largely on the global health side on molecular diagnostics. So he just asked me to share some things about the project and then also give a little bit of context for molecular diagnostics. Are most people in the room in science and engineering or have experience there? Okay. Most people on Zoom as well are more of the, STEM side of things. Awesome. I won't go too much into technical details, but for people who are interested, I'll touch upon it. I believe there's an hour for the class, so we'll spend the first part on giving context for the problem of access to diagnostics. I'll provide brief background on what are molecular diagnostics, and then also introduce a Prakash Lab project called D-Tree, which is a electricity-free molecular diagnostic device. And then we will, at the end, cover a little bit about distributed biological manufacturing for diagnostics, what that is and why it's important. I did not practice this talk, and so I don't know how long it will take, but if there is some extra time, then I did put a few extra fun slides at the end that we can go through if there's interest. And then part two, Anesta, who I think you all heard from her last week, is going to come in and talk about rapid prototyping of diagnostics and show some examples and versions of the D3 diagnostics. I think the takeaways are not necessarily specifically about molecular diagnostics, but more so I wanted to show you the thought process behind identifying unmet needs and then designing solutions for those. And I think that as you all are starting to think about your projects, gathering your teams, this might be a good case study / example for you all. Okay. To start with context and setting the scene is we recently went to Nigeria this past summer, and so I'm going to share that first and then backtrack more toward the basics. This is in a schoolhouse in Nigeria. We were setting up for a pilot study, testing a couple of different diagnostics that came out of the Prakash Lab and then also from medical students in Texas. And so this is what we were working with. It was an empty schoolhouse. We brought the chairs, we brought the tables. Someone literally rode in with the tables strapped to their back on a motorcycle from the city to this rural area. And so this is kind of lets you know that we're not working with a whole lot. And so when we don't have a whole lot, then what do we need to bring? Something that's very portable. All of our supplies and materials for our pilot study were just in normal suitcases and duffel bags that we brought all the way to the field site. And when we were going to the field sites, I think if you ever get a chance to go, you'll quickly realize that these rural areas are not easy to access. So if I can play it, this is a video that I was taking on the way there. Sorry, resolution is low, but essentially the roads that we were taking are not paved. They're super bumpy. There's lots of obstacles. We were in a huge van with maybe 20 people, but it was like a 10 or 15 person van. So we were packed inside and our car actually broke down on the way there. So oil was leaking out. It hit a sharp rock or something like that. So I think just basically it's not very easy to get here. And if we're thinking about healthcare delivery, then it's even harder to bring the tools, devices, materials that one would need to provide quality healthcare. Yeah.
Where in Nigeria were you?
We were in southwest Nigeria, Abeokuta, which is about like hour and a half from 2 hours from the capital.
Yeah.
Is that a sound for something?
I don't know. I think you're
Okay. Anyway, so access is hard. And so if access to the communities is hard, and also we don't have access to point of care diagnostics that can be used at site at the field, then how do we think around it? There's this great program called Riders for Health that used motorcycles to transport samples from rural communities to centralized city areas that had either laboratories to do the testing or had research labs who could process the samples. But the issue with that is the turnaround time could be anywhere from a week to a month or even longer, just because they're riding hundreds of miles, hundreds of kilometers across the country to bring the samples back and forth. And then you might notice this motorcycle, this has a cooler on the back of their motorcycle, and that's because they were transporting Ebola samples. So Ebola is, for people who don't know, is BSL-4, like highly pathogenic disease. And so to just very casually be bringing it on the back of your motorcycle is something that's not optimal. But at the time in Liberia and other countries in West Africa, that was kind of the only solution. So it's always good to, whatever device you're making, is to root that in a disease. And so today I'll talk just a little bit about schistosomiasis. Have you all talked about schisto in here with plankoscope or anything? Okay, so that's okay. Schistosomiasis is a waterborne disease. This is the parasite, the schistosoma parasite here in the corner. So anytime you go into the water, you have some sort of skin contact with infected water, then this parasite can go into your skin and into your bloodstream and cause all sorts of downstream complications, from just the normal symptoms like fever, vomiting, stomach aches, all the way to getting cancer. So it's very important to detect this disease as early as possible and treat it as early as possible. It affects hundreds of millions of people. Large majority, about 90% of schistosomiasis cases are in Africa, but there are still no easy to use field-ready diagnostics for schistosomiasis. So a lot of the current work that we are researching and developing is centered around schistosomiasis, but then it could also be applied more broadly to other diseases. You don't have to look at every single part of this slide, but this is just an overview of our workflow, and to show that even in that empty schoolhouse, we're still able to set up and take down every single day, drive over 2 hours to go there, and test 4 different diagnostics for schistosomiasis, and collect, process, diagnose 418 patients. And so, oh yeah, and I also wanted to show that the prevalence of schistosomiasis in this area of rural Nigeria is very high. We visited 5 different communities, and in 3 of those communities, about a third, or 1 in 3 people, had schistosomiasis. So this is definitely a disease that is highly prevalent, largely ignored until it's too late. And so this is just the outside of the schoolhouse that I showed you earlier. And I think the question that I'm asking here of, can we diagnose someone under a tree? Can we diagnose someone just out in the open, no laboratory infrastructure? The answer to that is yes, but then how do we go about thinking about that? How do you go about diagnosing someone without the normal healthcare facilities that you would think of when you're trying to test and treat? So with that, let me go a little bit into types of molecular diagnostics. There are a lot of different types. I'll talk briefly about 3 and then go into depth about 1. because molecular diagnostics are all about just detection of molecules. Those molecules can be DNA, RNA, protein. So anything that allows you to detect those microscopic items or components can be considered a molecular diagnostic. So one that you might be familiar with is the serological assay. Most of the rapid tests, like the COVID rapid test, where you did a nasal swab and then you put the drop onto this part here and it's lateral flow assay, that is a serological test because it's detecting an antibody. And there are other tests called nucleic acid amplification tests that are detecting at the DNA or RNA level. And so that's what we'll go more into today. And then the third type that's gaining more and more relevance is sequencing. It used to be that sequencing was the super hard thing to do, but then now with technology advancements, then sequencing is becoming a whole lot easier. So that's also detecting at the DNA level, but you're actually reading the individual base pairs. Whereas something like a nucleic acid amplification test, you're just generating many copies of that DNA and detecting the number of copies.
I have a question.
Yeah.
So excuse my ignorance, but when you're detecting antibodies, and these are the antibodies that you produce as a response to the infection?
Yes, yeah,
Okay.
yes, yeah. So that's exactly what this graph is showing, is the immune response to a certain pathogen. This is a graph that I pulled from a paper that was reviewing some sort of viral pathogen. And so they call it viremia, like level of viral particles inside that you're infected with. So this is level of viral copies that you have, but then the black lines are the amount of antibodies that you have, so immunoglobulin antibodies, and that's your immune response. And what I want you to notice is that there is a delay between the onset of sickness, onset of pathogenesis, and your body's response, right? That it makes sense, right? Because your body needs time to generate these IgG antibodies. And so for the COVID tests, the normal rapid COVID tests are detecting IgM and IgG antibodies, but they're only able to have sensitive detection a certain number of days after the virus is already incubating in your body. And that means that there's kind of an opportunity here from the onset of pathogenesis to kind of peak virality to have an earlier detection. Yeah.
May I ask, some people don't generate antibody responses.
Yeah.
In that case, the test fails, right?
Only the test would fail. Exactly. So there are certain populations like Jesse just mentioned, where if they don't produce an antibody response, then it's potentially very dangerous because the normal diagnostic test will not be able to detect it. So if you remember people who had autoimmune diseases or were immunocompromised during COVID-19 had to be extra careful. Some of them never left the house just because of things like that, where you're not able to properly assess and treat the disease because you don't have the proper immune response. What's next? Okay, so how do we go about having earlier detection of a disease? One way is through molecular testing or nucleic acid amplification tests. I'm going to talk about two. The first one is called PCR, and then the second one is called LAMP. So first one, PCR, polymerase chain reaction. If you all were here when Stanford was still doing the testing and you would do the nasal swabs and then send them to a lab. So that lab was doing a nucleic acid amplification test. With PCR, you're basically generating many copies of your target DNA. This DNA can be schistosoma, parasitic DNA, or you could have your SARS-CoV-2 RNA, and then you have to do reverse transcription into DNA and then follow this process. And what it's basically doing is it's generating 2 to the n copies of your target DNA, where n is the number of cycles that you're going through. The thing about PCR is if you are running a typical PCR reaction, it'll take a couple of hours, 2 to 4 hours, and you need to be a trained technician to run it because you have to do sample extraction and processing and then also run it on a PCR machine. There is good specificity. You have these red pink parts that are called primers that are specific to your target pathogen, DNA or RNA. And then with PCR, there's a couple of different steps that you have to go through per cycle, where you first have to pull apart the double-stranded DNA, and then you lower the temperature to allow the primers to anneal to your now single-stranded DNA, and then you raise the temperature back up so that the DNA polymerase can then extend that strand. So the thermocycling process has to be very precise, and that's why we need bulky equipment to do that. If you've ever seen a PCR machine, it's quite big, it's not very portable. Now there are more portable options, but typically in a laboratory setting, you're going to see kind of this big chunky equipment sitting on the bench top. The second nucleic acid amplification test that I want to talk about is called, the acronym is LAMP, and it stands for loop-mediated isothermal amplification. You don't need to follow all the details, but what's very cool about this is instead of 2 primers, you have 4 to 6 primers, and these 4 to 6 primers can target 6 to 8 different specific locations on the DNA, and they make these cool, like twisty, turny, dumbbell-shaped products that are able to prime themselves. So essentially, you have a higher efficiency reaction because you're generating more copies in the same amount of time than you would in PCR. And LAMP tests are also much faster. You don't have the same sample processing requirements as PCR. They have higher specificity because of those 4 to 6 primers. And you can also run these tests isothermally, so at a single temperature, which means that you're reducing the equipment requirements because you can run it in hot water. Yeah.
Like for this type of test, is it like more expensive to get the equipment, or is it like around the same price as like a PCR?
Oh, okay, so I think that is a great question, just because you can run a LAMP test on a PCR machine, but then also you could run it just in a dry bath. You can run it in a water bath, and then later on, the test that we're developing is run just in a cup of hot water. So there's many ways to innovate around the equipment just because you now only need to maintain the temperature at 1 temperature rather than cycle through 30 to 50 cycles through 3 different temperatures, which requires a lot more precision. So you kind of predicted what the direction that I was going in is, well, LAMP typically requires less bulky equipment, but are we able to have no bulky equipment? Are we able to get rid completely of that equipment requirement? Answer is yes. And so I'm just going to flip very quickly.
Hey,
through these.
Hope, before you continue, I had a question on the other slide. This is Camille Gaines on Zoom.
Yes. Hi, Camille.
Okay,
Go ahead.
going back to the LAMP. I've never really done a LAMP, but I'm familiar with like library preparation and PCRs and all that. So, quick question on the LAMP for the isothermal portion of the reaction. How do you know what temperatures, like for depending on what type of like product you're looking to get? How do you know what temperature? Is it like a trial and error type thing, that you do with PCRs, or is it something a little bit more standardized?
Yeah, that's a great question. So what's not shown here in this diagram or schematic is that all of this extension, all of the synthesis of the DNA copies is done by a enzyme. It's done by a protein, a DNA polymerase that comes from a thermophilic bacteria. And so the temperature is largely determined by the temperature at which that DNA polymerase is active. Did that
Okay,
answer
thank you.
your question? Yeah. And so the DNA polymerase that we're using operates about 63 degrees Celsius, but then if you go any higher, the protein has the chance of denaturing and becoming inactive. If you go any lower, then the reaction may not happen at all, or it may just be super, super slow. So there is a sweet spot that you can do some testing around. And then we're actually trying to engineer a new DNA polymerase that's able to operate at higher temperatures because then you have faster kinetics. This is the D-Tree device. It's version 1 of the device that Anesta, Manu, and many other people during COVID came up with the design and actually produced it. So the inspiration is from ballpoint pen manufacturing, and you're only using that cup of hot water to run the test. I'll show a workflow with the updated device in a few slides, but this was from COVID, and this is the part that Anesta is going to cover when she comes in later today about the rapid conceptualization to actualization. How they described that time was super chaotic, and they started with a design in the news kitchen, and then 3 months later they had an injection molded device that they were doing 600 to 800 person usability studies on. So it was an incredibly stressful time. I was not there, but I think it is possible when you have a pressing problem and have the urgency to rapidly design a solution to it. So Nesta will talk more about that. And the device itself, there's many design criteria for the device. You can, maybe think of a few where the parts need to be easily injection molded or easily attainable. They could be 3D printed. They could be found just at your local market or store. So things like that is what they were thinking about, especially since there were supply chain shortages during COVID-19. And then they also ran usability studies in both Guatemala and the Bay Area. And so these were studies where the participants did not have in-person training, but just looked at the instructions for use, or IFU, and were able to successfully operate the device. So the device also needs to be easy to use, in addition to not requiring any sort of expensive equipment, and in addition to being cheap. So there's many design considerations for when you're talking about frugal science and frugal technology. What do I want to talk about here? Okay, so in the case for schistosomiasis, we are sampling urine. We're taking urine samples from the study participants because in the urine samples, there are schistosoma eggs, and then inside the eggs, there's genomic DNA that we're trying to detect. So if you have the genomic DNA from your schistosoma egg, then you use LAMP to amplify it and make many, many copies. And how do you visualize that? Well, we can't do any sort of optical readout without some sort of external device. So the way that we think around that is, how about we just have a color change where just by eye, you're able to see the difference. So that before the reaction, you'll have this pink color, and then after the reaction, it'll turn yellow if the genomic DNA is detected, and then it'll stay pink if it's not detected. I showed you a few slides ago the first version of the D-Tree device, and that was a version that was designed for COVID-19, which used saliva samples. But then for schistosomiasis, we're now using urine samples, which is a much higher volume sample, and so we have to add a filtration step and then also change the design. There are many things that we had to consider for the updated version, and then Anesta will also talk a little bit more about that later. But there's now a version 2, but also many sub-iterations between all the versions. Yeah.
Changing the color.
Yes. So I don't have the mechanism here, but it's a good question where we have this dye that's a metallochromatic indicator, which means that it will change color based off of the concentration of a certain metal dye inside. And so in our case, the dye that we're using will detect zinc metals. And then as the reaction proceeds, the zinc metals are pulled out of the reaction from pyrophosphate, which is a byproduct of DNA synthesis. And as the zinc ions are pulled down and are no longer free in solution, then the dye will change color to yellow. So the dye complex, this is probably more detailed than you want, but it's a complexometric dye, so it will form a complex around the zinc metal ions. So in the complex form, the color is pink, but then when there's no zinc ions available and the dye is just floating free in solution, then it turns yellow.
And the zinc is sequestered when you amplify this specific DNA.
Yes, so zinc is sequestered. Great question. Zinc is sequestered because when you're synthesizing DNA, you produce a negatively charged byproduct. And then so the zinc and the negatively charged byproduct then form an ionic bond and then are pulled out. Yeah.
And how do you know it's the schistosomiasis that's doing replication?
How do you know if we're detecting sorry.
How do you know it's not just like any random?
Yeah, so great question. There are something called primers, which are short oligonucleotide sequences that specifically bind your target DNA. So without the primers, the enzyme is not able to start its synthesis. It's not able to make copies of your DNA.
Okay, so you give it.
You give it, yeah, kind of like an instruction of this is exactly what I want you to target. And if it's not there, then you're not going to have the copies of DNA generated, and then you're not going to have the color change. Okay, I didn't realize it was so late. Hello. Okay, so this is kind of with version 2 of the device where we add the sample and then close the lid, and then the sample is incubated inside of a cup. This is kind of like your standard stainless steel insulated cup. And that first incubation is to break open the eggs, the schistosoma eggs, so that the DNA is released in supernatant. And then you do a couple of like tapping transfer mechanisms with the valves, take photos, and then you have a second incubation at the 63 to 66 degrees Celsius for about 40 minutes. And then when you take it out, there should be a color change. These are where the reagents are housed. This was just a trial run, so you don't really see a color change, but if you had a positive sample, then you would see a yellow color, and then if you have a negative, it would stay pink.
How do you store your reagents? How do you bring them to the field of the NTP?
Yeah. So that's something that Anesta is working on right now, where currently the fresh or wet version of reagents requires storage at -20 degrees Celsius. And so we have a couple of different options for lyophilizing the reagents, which means that we're removing the water out of it. And then once there's no water, there's no medium for, it's mainly the enzyme that's most sensitive to this. So then there's no medium for the enzyme to become active or denature. And so what we're hoping to bring to the field is a lyophilized or shelf stable version of the reagents. Yes.
How do you dispose of the sample after?
Yeah. So it's actually a very good question, because our current route is we have a nice waste stream process where we have just like a red biohazard bin and we just put it in, and then someone takes it to incineration. When we were in Nigeria, there was no waste infrastructure. And so we just asked the community to just burn the trash. And we don't yet have a good solution, like a better solution for that, but that is very standard for places that don't have like a garbage truck every Thursday to pick up the trash. They will just burn their trash. But yeah, that's the
By that point,
everything.
no longer infectious.
Yes, it's no longer infectious. The device is designed such that once you put in the sample, then you never open it back up again. But we still want to make sure we have a dedicated waste disposal process for it. So thank you for bringing that up. How much time do you want?
Oh, that's fine. It's more like a show and tell and like design question.
Okay, so this is the last section of this portion where I just wanted to introduce you all to distributed biological manufacturing, what that means and why it's important. So earlier we saw the riders for health. They have to ride from like probably the capital city, hundreds of kilometers to rural areas, pick up the sample, bring it back. So that's a centralized model where everything is in a large city. You have long turnaround times. It's very costly to run something like that. Whereas what we want to do is kind of shift that device delivery problem toward a decentralized model where in the rural areas at the site where the need is the highest, we're able to diagnose someone, we're able to produce the diagnostics that can then diagnose someone. So the benefits of that, there's a faster turnaround time, and then it also decreases the cost because you don't have the shipment requirements. And then you're also producing a very low cost device. So this is the first time I'm bringing up this example where if you think about distributed manufacturing, what does that mean? Well, if you want to say you want to buy an apple, then you need to go all the way to the store, like Trader Joe's, to get and then come back and eat it. But what if you grew an apple tree in your own backyard? Then you just need to walk 30 seconds outside, pick an apple, and then have it right away. So that's kind of the very simplified idea of distributed / local manufacturing is that the devices that you want to make are produced at the site of need. Well, okay, maybe not. Okay, I will go over this just because I want you all to know is that the enzymes that we were talking about earlier are very sensitive. They're probably the most sensitive to what's called denaturation. With your protein, it's normally in a folded state. The folded state imparts certain functionality to it, but then at higher temperatures, then that folded protein denatures. It doesn't maintain its original structure. If it doesn't have that original structure, then it's no longer functional. That's why if, say, you have a diagnostic that you want to ship from the US to Africa, you need something called a cold chain. So that shipment needs to be done on usually dry ice or have some sort of temperature controlled facility for the shipment. And then also, once you deliver the diagnostic, that diagnostic still needs to be stored in a facility. And that's very, very hard because many of the locations that experience high burdens of diseases don't have electricity. So you don't have a fridge, you don't have a freezer. I'm going to skip that. I think the basic idea for distributed biological manufacturing is that we want to be able to both produce and use the device on site or as close to the site as possible. One version that we're thinking of is kind of having a manufacturing facility inside of a shipping container. And that shipping container could be placed at more rural locations where they would then produce a device that could target schistosomiasis, but it could also be adapted for malaria, for tuberculosis, for other diseases that are of importance and high burden in that area. And what that would entail is both manufacturing of the reagents like the enzymes that we just talked about, as well as the hardware components. This is still version 1, but then the new version would be an almost entirely injection molded device. Yeah, so that brings me to the end of kind of the more theory qualitative part of the discussion. And I think that we can transition to.
Thank you.
Yeah.
What, sorry?
We're both on.
Yeah.
Do you want a Zoom link?
Yeah, or can I just send you a link?
Yeah. For the extra fun part, I was just going to go over some like fun ways to validate the diagnostic test using probability. And so.
Okay, what's your next steps?
For
this
after
project?
for the project? Yeah, okay, so great question. Next steps for the project is all about this protein, where we want to make this protein in-house. and make it thermostable enough that it can be stored at degrees up to 50 degrees Celsius, because that is now the new WHO standard with, climate change and rapidly rising temperatures. Then we need to be able to store these biologics at high temperatures. Okay, so we have a. Oh, no, it's okay. Oh, sorry.
Okay, cool. While we wait for that, so yeah, we have transitioned away from the first device, first versions.
Silence the camera.
Oh, yes.
Stuck. Yes. Okay.
This side.
Okay.
Yeah, so the version that Hope showed in some of her slides were kind of this longish thing with the blue cap and with straw-like element in the middle. That one worked for some time, and that one was designed very, very fast in like maybe 2 to 3 weeks or less. This was during COVID, and Manu just went to a Home Depot and got a bunch of tubes of various sizes. We played around with various ideas. Like at one point, I don't know if you're familiar with the paperfuge. You guys, so a lot of the sample prep typically required centrifugation to extract the DNA. So we at one point we had like a device that looked like this, where you would like put in the sample and then fling it like like a paperfuge, and we realized that was quite dangerous. So we moved away from that entirely, like within like 2 days. So then that's when we were thinking, okay, how can we move? Basically, what we have to do is move a certain amount of liquid past another chamber and past another chamber, like sequentially. And centrifugation did that, but what other ways? So instead of a centrifugation, it's more of a mechanical push, and that's when we had the kind of the plunger system where we had first the larger volume being pushed into the smaller kind of tube-like. And then is this shared already?
Oh, no, but
I just
can go
wanted
to
to-
the slide you want.
Yes,
and I can share.
So yeah, then I'll just kind of walk you through what went wrong in that one and how can we make it better, since that one was very, very rapidly designed. So there's version 1, as you can see. And then we went through this whole phase. As you can see, the date went from 2020 to 2025, so it's in 5 years. Version 2 was when we were trying to do like, how can we think of this very, very differently? We were just really stuck on this form factor, and that we couldn't really move beyond it. So this is just kind of like design exploration. There's no really physical prototype on this. But then from that, fast forward 2 more years, then we realized, there's these things. So we were playing with syringes. I just wanted to show you some of the old model. I don't have a syringe here, but there's an image here. So yeah, so there's this, syringes, but there's this element called a stopcock valve, which basically is, in simplest things, there's a cylinder in the middle that you can turn either toward the direction of the syringe, or you can lock it, and then nothing else will pass through, essentially. Or you can what we realized is we can actually use this mechanism because we realized it was pretty seal proof, like it was leak proof. So we can, if we imagine like another axis kind of running, the opposite direction of the syringe. So you can essentially pass through liquid and then capture it in the middle in that blue thing with that cylinder, and then turn it and transfer it another way. So in our device, we are transferring small, I don't know if you spoke about the volumes, but these are very small volumes. So I don't know if you guys can guess how much each of those little chambers can hold. Like where the dark color is. So many. For a reference, like the little Eppendorf tubes that you usually get, those are like 1.5 mils. So this is, microliters. So this is about 25 microliters. And it is critical that you transfer an exact amount because the reagent stoichiometry is based on that. So the reagents that are already stored in the device are meant to hold 25 microliters of sample and liquid, so when it's reacting, it needs that volume. So how can you transfer, when you're thinking about urine, someone would urinate, like 100 mil, right? And you only need like a subsample of that. In addition to that, there are these for schisto. So for schisto, we are trying to capture the eggs in the urine. So then, if you're just taking, let's say, 20 microliters from this big pool of urine, you might not capture the eggs, right? So first you have to filter the urine so that you have a much smaller volume and that you do capture the eggs. So the way you do that is with a it's all here. So this is the syringe idea. So we were like playing with this. So this is the diagram of how it works inside, essentially. And then we tested this, putting it in 37C to make sure that it does also pass the evaporation test. For the most part, it does. I think you can, play around with it a little bit more to make it even more foolproof. Like this is not in a bag. This was just like so 37C. And then we're looking at like what are these materials made out of. So as we speak, they are making the parts now. We are getting them shipped out tomorrow. So like we said last week, the injection mold cost a lot. So that took a lot of effort just to get it to the final design before they can actually make the mold. So I'm really hoping those things work next week. I'm gonna skip over this. It's just a little bit minute detail. But yeah, we were experimenting like different size valve, how much volume can we hold in this, how many chambers can we technically hold in this, because all of this will be connected in terms of the geometry of the final device. And we went through, even within version 3, several different iterations. So I want to just point out these 3 major ones. So for the first 3.1 is just integrating these geometry, right? So it's just understanding 2 plunger systems in a combined valve. How can we do that? And then there's a single plunger. And then the second version was looking at, okay, if I can push through one way, what if I can just use another plunger to push on the second way? So that was a kind of double plunger. And then we realized, we also need to injection mold this. So when we spoke with someone at Stanford, I didn't have proper training in injection molding before this, so this was kind of like learn on the go. But we realized that design here is not injection moldable for the various reasons that we kind of discussed last week, where it could not be pulled apart, it was too thick. There's various parts that are thicker than others, which meant that your geometry won't be stable. So if you look at any like injection mold parts, most of them, the thickness all around should be around the same, so that they cool and they melt and cool at the same time. So they don't like have one that's like sinking, they call it. So this is the filter. This is a very typical filter that you use in a 15 ml tube. So we were hoping that we could, stick something like this into the top of a 15 ml conical. These are standard, and then transfer that filter over. So the idea was that if you can transfer a filter directly, so if you have it on the 15 ml tube with the cap alone, you can hold it. So then you're just transferring the eggs. I don't need the whole 100 ml of urine. I really just need the egg and like a little bit of urine just for the human control. Yeah, do you guys have any questions yet so far? I know it's put out a lot of things. So you need a little bit of urine for the human control, but mostly we're looking for the eggs. So if you pass through, let's say like 100 ml, this is missing the filter, but just imagine there's a filter that looks like that white sheet in the red piece, where my finger is. Basically, you're just floating like a sieve. You're just capturing the eggs. So it doesn't matter how much volume you have. In the end, we will have 1 ml of just water in the device, so that will just resuspend the eggs so that the eggs can be lice. So once the eggs have been transferred, you close it, you put it in your cup, lice it with the hot water. And from that point on, the eggs have disintegrated, the shell have opened, now the DNA is exposed. DNA is much smaller than the filter size, so it can just flow through the filter, so it doesn't matter that it's on the other side of the filter per se, it will go through. Then from that point on, you use this as a transfer mechanism. So in this scenario, we have a multiplex. So this is a even much later version than the ones I'm showing here. This is the injection mold part. But yeah, to get from here to here, basically there's a huge flattening out, you can see, right? Basically, it doesn't need to be a cylindrical for any reason at all, but for injection mold, it's just much easier to have two things that are just kind of top and bottom and a cavity. So that's kind of what we did. We also, if you notice between this version and the one I'm holding in my hand, is also missing the two plungers. We realized we actually don't need those two plungers anymore. We can either create a vacuum chamber in these chambers themselves, so that by the time it sees liquid or anything higher pressure, then it will just transfer over.
No,
So go ahead. Or what we're doing is literally just temp it. Works really well. So like 30 seconds, I can temp it and it will just transfer the liquid really well. So all this pink side here, what you see, don't worry about the color. I'm just referencing the chamber. Basically, you can see that now everything is aligned essentially, right, from top to bottom. So if I tap, if that liquid there, it will go past. So why? Does anyone have any reason, any guesses on why we have to have this little bit of extra? This is not where the reagents are stored. The reagents are stored on the side.
then what's the red thing?
It's not reagent, so just imagine it's empty. So at the beginning it is empty, but we do need that chamber. Does anyone have any ideas?
The control.
No, so remember we have to capture exact volumes, right? So the exact volume is determined by this inner cylinder. The thing that rotates, right? This valve that rotates. This is like resin, so it sticks. Essentially, it rotates, right? Like this. So that captured volume essentially is 50 microliters because I'm duplicate. I have double chamber, so 50 microliters. Now if I tap, I want to make sure that I fill exactly that and all the way that. So then I make this as an access chamber. So it has to at least go through that and fill the entire of that cavity. Otherwise, if without that, then it's hard to fill all the way because of air cavity, air pockets. This just allows it to be easier to exchange air and liquid so that it could fill up entirely. And then from that point on, you just turn it to the first set and then same thing, tap it out until it's empty. And then you can do the second multiplex and then tap more sample in and then transfer onto the other one. Essentially. And then once you have transferred both, that's when you lock it back. So now what you want to lock is the two reaction chambers, like the two pairs of reaction chambers. And then from that point, you just dump it in the water for 40 minutes, and then you'll see your results from here. Yeah, but I just wanted to share kind of the whole design iteration and that it's not a like, we woke up one night and we just like, oh, this is the one, there are like various ones that we tried. These are like the larger valves that we also tried. Yeah, so we initially started with just like the basic, like the PLA based printing. So this is kind of like, oh, how do you do a plunger in a plunger if like you don't accidentally push the other side? Anyway, there's a lot of different exploration, but yeah, feel free to look through it. And then at one point we did this the first time we flattened, or the very first time we flattened it, and you can see that the walls are much thinner, and I didn't realize you can easily break that in half if you're trying to turn this valve. So that's when we introduced the higher lip so that it can be more structurally stable. But yeah, feel free to look through any of this. Some of them still have the double plunger. Some of them have the single plunger. This is when sometimes I'm just trying to test one element. So like say the cap was like not sealing, so I don't want to print the whole thing, or I just want to test the cap. So sometimes that works too. Like for example, see, I'm just testing the top of it. Yeah.
It's inside the device.
Oh yeah, so when it comes in as the components, so we have the body and this part is removed. So before putting it in, then I would put in, oh, sorry, I have to backtrack. You didn't talk about the capillaries at all, right? So for the wet reagent, we used to store them in capillaries. So you can also see capillaries, like literally just glass capillaries stored in the device. We had that like this too, where we wanted to do a multiplex class 1 chamber, but with 3 distinct capillaries in there. We tried that. The reason why we are moving away from that model is that we realized because the capillary will essentially be only sealed if it fits that circular geometry, right? If for some reason you're in this curved part and your capillary is up to here, now that's not sealed. that has the potential to jump across the two capillaries. So we realize that's doesn't work. If we want to completely multiplex, we can either do two of the same one, which we're doing here, and then two of the same one across the other side. So that even if they jump to each other, it's just like multiplying the reagents by 2. But we do store it in the capillary. So when we lyophilize, we also lyophilize in the capillary. So we try to make the volume so low so that when we, like in terms of the water content, even before the lyophilization, so that the dry content is very small. Ideally, this would be in a pellet. This can be done in a company, just not right now, like not in Stanford by us. We need like different mechanisms for this, but right now for what we can do for our testing, we can dry them like as small as like 2 to 3 millimeters worth of capillary. Now there's a way to do it. That's something that Hope and I have been experimenting with. If you ever try to lyophilize something that small, you will never ever test just doing it like that. Because first, the process of lyophilizing is you have to freeze it in -80 and then put it in the lyophilizer to do sublimation to remove the liquid. If you are freezing that tiny volume, the mere touch of it with your finger can just instantly melt it. If you melt it, then your whole lyophilization just doesn't work anymore. So what we've been doing is starting with longer capillaries, but having a tiny volume, so then it can still travel across during the vacuum process, but then still stay within the capillary, and then we chop off the rest of the capillary thereafter, and then we store them in the chambers.
So before you put your valve in, you would like introduce
Yeah, so
like frozen.
tiny capillaries that have the reagents, and then I stuff them in, and then snap fit this, and
It's like a powder. It's dry.
yeah.
Okay.
Yeah, and then if you do it well, then they instantly dissolve upon meeting a liquid.
I think we're about time.
Yeah, so let me know if there are any questions, but yeah, we have some ridiculous prototypes. Kind of like, I don't know what I was thinking. Yeah. Yeah, but these early ones were actually really fun. That's when you realize you get the confidence that something like that might work.
Yeah.
But you see, this one we were still stuck on the geometry. So sometimes you have to just like erase everything and like from your memory anything you know about the project.
Sure.
Yeah, otherwise you'll just stick with this cylindrical format all the time, and it's just not even needed. Like nothing says it has to be cylindrical.
Thank you.
Yeah, thank you.
I mean, it's expressed to see things like that. If you're delivering somewhere, let's say that box falls over.
Yeah, I mean, other than like us dropping it on the ground.
Yeah.
So we did that with all the rest. But I think we're gonna try again with this next time. Once we get past all the systems. So these are also resin. The first thing before stress testing is, obviously we have to do the essentials and compatible. It's sort of. Yeah. Because it doesn't work when it has material. But yeah, we did. Yeah, we did. I asked what part of the stress testing. I was a bit technical. That's something you have to drop it. Levels 1 meter and then it's not great. Then you have to test, okay, you're into the sugar grade. Other stress tests is also the heat.
So
Then you have more in like boiling water does it more. So that's something that we also test. So we go with big time. So they did a lot of that stress testing for us. So yeah, they made sure that the dimensions are still the same, close to 95. So yeah, this material is not, so this melts. Yeah, what about it? But yeah, this will like warp. It like warps in 63°C or above 41.
Yeah, okay, I see. Yeah, wait, that's a cool. Yeah,
Yeah.
thanks for sharing. Yeah.